Dear friends, in this video I am going to talk about telomerase, an enzyme which is responsible for the stability of telomere, that is, end of the chromosome. Telomeres are the end of chromosomes. The two ends you can see here clearly. Telomeres are the genetic material at the end of chromosome and are essential for proper chromosome structure and function. These are repeating sequence of base pairs. In human and all vertebrates, the sequence is TTAGGG. The sequence forms structures that protect the DNA from attack by nucleases that degrade the ends of DNA molecules in the cell. Without these end caps, chromosomes sticks to one another and undergo structural changes. These activities threaten cell survival and the faithful replication of chromosomes. Chromosomes slowly lose base pairs at the end of their strands during replication. To overcome the end replication problem, most eukaryotic cells utilize the enzyme telomerase that allows the replication of lagging strand to be completed. Telomeres shorten as cells divide. After each replication, the segment or the size of telomere slightly reduces. Telomere shortening leads to cell death. How it happens? Because once the cell chromosome starts losing its telomere, the consistency or the protectivity of that particular chromosome decreases and the stickiness leads to breakage and reunion, and that leads to cell abnormality, and finally the cell death will occur. Telomerase was discovered by Corel Grieder and Elizabeth Blackburg in 1984 in the ciliate Tetrahymena. Tetrahymena are free-living ciliate protozoa that can also switch. They can be living in fresh water. Tetrahymena species used as model organism in biomedical research. Grieder and Blackburn was awarded Nobel during 2009 in physiology or medicine for their discovery. Telomerase is a ribonucleoprotein enzyme capable of extending chromosome ends consisting mainly of protein. It's a specialized reverse transcriptase that contains both RNA and protein and critical nucleotide template for building telomeric subunits. How does telomerase work? Telomerase place one strand of DNA on RNA, positioning itself so that the template lies adjacent to the tip of the chromosome. Then the enzyme adds one DNA nucleotide at a time until a full telomeric subunit is formed. When the subunit is complete, telomerase can attach another by sliding to the new end of the chromosome and repeating the synthetic process. This is role of telomerase, how it acts at 5' and 3' ends, and it is also an active material in DNA repair mechanism along with ligase. Telomerase allows telomere length equilibrium maintenance. Like this. By the continuous activity of TERT, telomerase helps in length equilibrium maintenance. By hairpin loop structure, replication continuously. Lack of telomerase activity in human cells leads to telomere shortening. Eventually, this leads to genomic instability. For continued cell division, telomere loss must stop and telomerase must be activated in the cell. These immoral cells are important in two areas of research: that is cancer research. There are many different types of cancers, varying appearance and locations in the body. The unifying aspect of cancer is uncontrollable growth and immortality. The tissue expands without limit, compromising the function of organs and threatening the life of the organism. Cancers arise when a cell acquires multiple genetic mutations that together cause the cell to escape from normal control on replication and migration. As the cell and its offspring multiply uncontrollably, they can invade and damage nearby tissue. Lack of telomerase would retard the growth of tumors by causing continuously dividing cells to lose their telomerase and to die before they did any damage. Therefore, removal of telomerase could be a possible treatment for cancer. In regard to this theory, many experiments have been conducted to detect telomerase in cancer cells. Telomeres and aging. The one aspect was cancer and the second is aging. Cellular aging is a process by which a cell becomes old and dies. It is due to shortening of the chromosomal telomeres they reach critical stage. Cells that cannot reactivate their telomerase will stop dividing or senescence. Ability to make body cells live forever using telomerase research could be important in anti-aging process. Thank you. Hope this video is informative for you. If you liked, kindly subscribe my channel. Thanks.